All right. Thanks everyone for joining us today. It is my pleasure to introduce Omer Dushek. Omer is an associate professor from the Sir William Ying School of Pathology at the University of Oxford. His research focuses on studying the functionalities of T cells in regulating immune responses using a combination of quantitative experiments and mathematical modeling. So today he will share with us some of the related work about the ligand discriminatory power of the T cell surface receptors.
Okay. Thank you so much, Ying. And thank you for inviting me to give this talk. I actually haven't given a talk to a mathematical biology audience in a while. So I've given a talk to sort of applied mathematicians and to immunologists. And so I haven't had this kind of target audience where people have some biology knowledge. But I gather that it's going to be quite diverse. So I'm guessing, and I've met someone studying, for example, neuroscience. And so I'm going to try to give a bit of a broader background than I usually give. But I think the best thing to do is if you have any questions, just to stop me at any point and ask, so that we're all on the same page. Because I think understanding the background is going to be important for understanding the kind of work we do in the math, some of the mathematical modeling that comes after. So I did my undergraduate degree in physics, and I did my PhD in applied math. And it was really near the end of that PhD that I started to get interested in experimental immunology, and in particular to study the T cell receptor. And so for my postdoc, I decided to actually go into an experimental laboratory, actually here at the Dunn School of Pathology in Oxford. And it was during my postdoc that I started to do some experiments, and that sort of gave me the confidence to maybe try to start an experimental lab. And so my lab right now, as you'll see, does quantitative experiments, and then we fit mathematical models to the data that we get. And that's kind of where the novelty comes, not so much from the experiments, which immunologists have done for many years. It's really from doing them quantitatively and fitting models and inferring parameters. So the cells that my lab studies are called T cells. They're very important white blood cells, and they continuously recirculate in your body. So unlike most cells that kind of stay put in one place in your body, these cells travel around. They leave your blood and go into tissues. From those tissues, they drain into local lymph nodes, and then they could go back into the blood, back through tissues, and do that cycle over and over again. And the reason they do that is because their job is to continuously scan the surfaces of other cells for what you might call the molecular signatures of infections and cancers. And we call these molecular signatures antigens. So here's just a little movie showing you a T cell crawling around. In red is a protein called actin that's labeled. And I'm just showing this just to highlight how dynamic their movement is. And here's an example of a T cell scanning the surface of another cell. And it really kind of pushes and probes on that other cell membrane. And the reason it does that is because what it's trying to do is get a protein on its surface called the T cell antigen receptor in very close proximity to its ligand, which is the antigen molecule, which I'll talk about in a few slides from now. And it's really through this interaction that happens at the cell-cell interface that the T cell will determine if that cell harbors any of these signatures, antigens of infection or cancer. And just in case you're interested, the distance that these two membranes have to come into proximity is about 14 nanometers. So it's a very, very tight junction that's formed between these cells. And so once the T cell recognizes the antigen molecule on the other cell, it can transduce signaling that could lead to different kinds of responses. And collectively, these kind of responses help T cells not only initiate, but also regulate adaptive immune responses. These responses, for example, lead to the production of antibodies by B cells. They lead to local inflammation, and they could also lead the T cell to directly kill the cell that it's interacting with. And so you can imagine that all these responses help T cells clear infections and cancers. But when this activation is inappropriate and the target antigen is not derived from an infection or cancer, it could also lead to allergies. This is when the antigen is innocuous. Autoimmunity, when the antigen is derived from your own self tissues. And it could also lead to the rejection of transplants because T cells recognize these transplants effectively as foreign. And so they're clinically very relevant, of course. This is just to show you that basically we've mapped out most, if not all, of the signaling proteins in T cells that relay information from the receptor at the cell surface to these different responses that they initiate. Many of them involve changes in gene expression, and therefore they're kind of downstream of transcription factors. And depending on how you count, there's probably at least 100 molecules involved in this pathway. And it's worth mentioning that this number is much larger than the number of molecules involved in pathways for other receptors. So other common receptors, for example, are cytokine receptors, where there's 2 molecules, or arguably 1 molecule between the receptor and the activation of a transcription factor. So things are a lot more complicated in the signaling pathway in T cells. Okay, I hope that's clear so far. I'm just giving you some background information. So I want to talk about the ligand. What is it that T cells actually recognize? So T cells recognize something called peptide MHC molecules. These are short peptides that are derived from proteins. And so this cartoon looks complicated, but I'll just take you through the essential features. All your cells, or at least the vast majority, all nucleated cells, have a process whereby whatever protein that they're producing, some fraction of them will get immediately shuttled into the proteasome, which is a structure that can basically chop up those proteins into short little peptides. They vary in length from about 8 to 14 amino acids. These peptides are shuttled into the endoplasmic reticulum, where they're loaded onto molecules called MHC, which are then shuttled to the surface of the cell. And the way that the structure of these MHCs, the way you should think about them is kind of like a hot dog in a bun, where the MHC molecule is kind of like the bun, and the peptide is the hot dog. Okay, and these MHC molecules basically present these peptides to the outside world. And the idea is that these peptides on these MHC molecules provide a snapshot of what kind of proteins that cell is currently producing. And so you can imagine that if a virus, shown here in blue, infects a cell, it's going to start to hijack the cell's machinery to make its own proteins, for example, this blue viral protein. And this blue viral protein will also be subject to this degradation mechanism. It'll generate small peptides loaded onto MHC molecules in the ER and then expressed on the surface. And so these cells, which we collectively call antigen-presenting cells, don't really discriminate between which peptides they put on their surface. They'll put peptides that are derived from your own proteins, and also peptides that are derived from foreign proteins, for example, from viral proteins. And it's really the job of the T cell to decide whether or not this cell on its surface has any of those foreign viral peptides. And if it has some, T cells have the ability to initiate an immune response. Okay, they could do many things, including directly killing this cell. But I want to make one point on the slide, which is that the majority of these peptide MHCs on the surface are likely to kind of have peptides, their hot dogs are going to be derived from self proteins, your own proteins. And that's simply because most of the proteins in the cell will be ones that are your own proteins, even if it's infected with a virus. A given antigen-presenting cell has about 100,000 different MHC molecules on its cell surface. And you could imagine that these antigen-presenting cells actually express very few copies of any of these foreign-derived peptides. And we know that directly from measurements. So for example, if you take cells and you infect them with HIV, you could actually count how many peptides, MHCs, they have on the surface, and the number is low, somewhere between 8 and 46 in this particular study. And that makes sense because viruses, as you could guess, they've evolved mechanisms to try to reduce expression of these MHC molecules so that they wouldn't be recognized by T cells. Does anyone have any questions on that? Is that clear so far?
I think we're good so far.
Okay.
Yeah.
Okay. So what I'm going to do now is just talk to you about T cells. Okay. So like we said, T cells have to decide which peptides are derived from self and which are derived from foreign. And you can imagine that unlike other receptors that have evolved with their ligands, T cells, they've been designed to recognize ligands that they've never actually encountered before. And the way this process works is quite remarkable. They start off as precursor cells in your thymus. And effectively what happens is that through gene recombination and insertions and deletions, they are able to generate a random T cell receptor. And in fact, this is an area where a lot of modeling has been done to try to estimate theoretically how many different T cell receptors can be produced. And it's about 10 to the 15 different receptors. And I want to show you where that variability is. So here's a structure of a T cell receptor binding the peptide MHC molecule here at the bottom. And in gray here is the kind of bun. And then the yellow here, I don't know if you could see it, is the peptide. And the T cell receptor uses loops at the bottom of its receptor to kind of touch the peptide. And those loops are highlighted here in different colors. And it's really in these loops where different T cell receptors vary from each other. And it's been estimated that you could have more than 10 to the 15 different receptors. And I think the estimates, the more recent estimates are 10 to the 20 or even higher. So you can imagine that if you're generating a protein, a receptor by random gene recombinations, it's possible that by chance you'll just get a protein that won't fold properly and won't be expressed. And so one of the things that happens early on in your thymus is that T cell receptors are screened for their ability to bind peptides that are expressed in your thymus. And because your thymus should be a healthy organ that's not infected, all of these peptides will be derived from self-proteins. And therefore, if the T cell receptor that's generated can bind to some of these self-proteins, it produces a signal that allows the T cell to continue development. If it gets no signal, maybe because the receptor is not folded correctly, the cells die by a process called neglect. And that's shown here by these kind of cells that don't look nice and circular anymore. So these cells die. The next thing that happens is that the cells undergo what's called negative selection, where T cells that bind too strongly to self, peptides, are deleted again by a process, by an apoptotic process. And so what this means is that T cells that are ready to leave your thymus and start to recirculate and look for potentially for foreign antigens have a kind of weak affinity for your own self-proteins. And so in your body at the moment, you have probably something on the order of 10 to the 12th T cells. They weigh about 500 grams, and they have about 10 to the 8 distinct T cell receptors. So some T cells in your periphery that have one particular T cell receptors might have divided several times. And this often happens after an infection, for example. You get proliferation of one particular T cell that has one particular T cell receptor, and therefore it has makes many copies of it. And so collectively, this 10 to the 8 receptors, we call that your T cell repertoire. And it's using this repertoire of T cell receptors that your immune system hopes to be able to detect some foreign antigens. Okay, so I'll just summarize by saying that T cells have to ignore normal cells that express only self peptide MHCs, but they have to respond to abnormal cells that express even small quantity of some foreign peptide MHCs that those T cells never encountered before. And these self-peptides, you can imagine, because they're very short in sequence, 8 to 14 amino acids in length, they can be very similar at the level of the sequence of the amino acid and also in terms of the structure that they form. And so when the T cell receptor sees these and binds to them, you could imagine that that binding can be quite similar. And so this was a big open problem in the field. How do T cells discriminate between self-antigens and make sure that they don't become activated, but respond to cells when they express even small quantities of these foreign derived peptide MHC molecules. And so an initial conclusion came in the 1990s. What people did is they took, it was actually all done in mice, and they took mice that all had the exact same T cell receptor. So they're called transgenic mice. So instead of having 10 to the 8 randomly generated receptors, all the T cells had the same receptor. This particular receptor, 3L2, happened to recognize a short peptide from hemoglobin. And so what they did is they took T cells, all these T cells, and they mixed them with these antigen-presenting cells that they loaded with different concentrations of the peptide shown here on the x-axis. And so you can see that for the hemoglobin peptide shown here in the black circles, as you add more and more of this peptide, if you measure the activity of T cell here by their ability to proliferate, you can see that it nicely induces the proliferation of these cells. What was remarkable was that they were able to find peptides where they make one amino acid mutation. And you can see here that this peptide with a single amino acid mutation doesn't activate the T cell, even if you increase its concentration quite dramatically, in this case by 10,000-fold. And this was reproduced in two other mouse systems, one called Touboul and one called OT1. This OT1 receptor is used really heavily in the immunology community. And in fact, it underlies most of our understanding of how T cells discriminate between these very similar ligands. And again, the wild-type peptide activated T cells, here they're measuring a signaling molecule called ERK in T cells. And they identified point mutations like this blue and red ligand that completely abolished the T cell response with just one amino acid mutation. And so this wouldn't have been surprising if this amino acid mutations just blocked binding between the ligand and the receptor, right? If there's no binding between these, then of course the cell is not going to be able to respond. But what was remarkable was that when they took these proteins, the T cell receptor and these peptide MHC ligands, and they made them as purified proteins, and they measured their binding affinities, what they found was that there were very modest differences. In this system, 2-fold, in this system, 7-fold, and in this system, 3-fold. And so you can imagine that if you wrote down a simple model of mass action between a receptor and a ligand, and you had two ligands, this red ligand, and another ligand that had an affinity that was just 3-fold lower, then in that mass action model, all it would do is predict that that red curve should be shifted to the right by 3-fold, meaning you'd have to have 3-fold more ligands to get the exact same T cell response. But that wasn't the case. You could see that in this case, you were able to increase the ligand by 100,000-fold, and still the T cell wasn't activating. And so as a result, what people concluded were the T cells are highly discriminatory to the TCR peptide MHC binding kinetics or affinity, so that these self-antigens, that the T cells are recognized in the thymus, must be binding with just a slightly lower affinity. But that slightly lower affinity, even 3-fold lower affinity, is enough to completely to not allow the T cell to become activated to your own normal tissues. If the T cell by chance happened across a foreign antigen, one that it's never encountered before, and it happened to have a slightly higher affinity, then it would induce T cell activation and initiate a global immune response in your body. And so because T cell antigen discrimination, at least according to these initial studies, was thought to be perfect or absolute, because ligand concentration couldn't compensate for a lower affinity, people refer to this kind of discrimination as perfect or absolute based on the affinity, okay? You couldn't compensate with a higher ligand concentration for a slightly lower affinity. And so, so again, this is kind of a mass action model. And in the mass action model, like I said before, if you lower the affinity, for example, by 2-fold, and I should say KD is a measure of affinity where larger values means a lower affinity. So biologists like to use this number, just in case it's not familiar to you. And so as you lower the affinity, you increase KD. In an occupancy model, the curves just shift a little bit to the right, linearly proportional to these changes in KD. And this is what's called an occupancy model. And this would be the case if the T cell was simply responding to how many of its receptors were bound. In 1995, Timothy McKeithen proposed this model. And he basically said, well, if there's kinetic proofreading at the T cell receptor so that when the ligand binds, it doesn't activate right away, but there's a delay, and only after a long time does it activate the cell, then the binding affinity or the lifetime of the interaction is going to be highly sensitive to whether or not you get signaling. Only interactions to stay bound long enough will trigger signaling. And this basically fans out these curves. And so when I was a grad student, actually, this problem was given to me because you could see that even with kinetic proofreading, we can't quantitatively explain that kind of perfect antigen discrimination that I showed you on the previous slide. And I'm being a little bit vague here, and I'll come back to this at the very end of the talk, but this model is basically insufficient to explain the data on the previous slide. And so when I was a student, it was sort of presented to me as a problem, a quantitative challenge in T cell immunology, which is what mechanisms must you augment this kinetic proofreading with to kind of get this absolute discrimination so that even small fold changes completely abolish the T cell response. And this is basically where the field was a few years ago. I would say maybe, I guess, a year ago. people were trying to augment kinetic proofreading with what I call mechanism X. So for example, people introduce feedbacks, cooperativity, something called catch bonds, and trying to invoke mechanics. I proposed that there might be some process called induced rebinding, and so on. And there's been a bit of a debate as to what molecules in the cell actually mediate some of these mechanisms. So this was the situation about a year ago. And about a year before that, I instead of trying to understand the mechanism and explain this data, I actually went back to the original studies in the 90s that reported these small differences in affinity. And to cut a long story short, some of that data is a little bit questionable. Basically, it looks in the data, if you look at it very carefully, that it's actually based on measurements using protein aggregates. And so what that means is that the twofold change in affinity may be very different if you did it with monomeric proteins, not big clumps of proteins. And so we set out to do in my lab is to try to reproduce these kind of measurements using more accurate measurements of affinity. And we also decided to do it with human T cells because people have kind of shied away from studying antigen discrimination with human cells. And so what I want to tell you in the rest of the talk is basically our new data, a kind of meta-analysis we did of some published data, what is discrimination for other receptors, and then a little bit about the molecular mechanisms and how we could actually fit this kinetic proofreading model to our data. Then I'll summarize. And like I said, if you have any questions at any point, just please stop me. So this work was done by a PhD student, Johannes Petman, and a postdoc, Anas Abu Shah, together with Anna Hun. And so all of them have a little bit of math and biophysics background, and they're all proficient in experimental biology. And a lot of the data fitting that I'll show you was done by Daniel Wilson, who is a postdoc, with Sam Isaacson, who's at Boston University, who's a long-term collaborator of mine. And all of the protein work was done by a protein biochemist in our lab, Misha Kuskov. So I'm going to actually skip this first part because it's quite technical and it's focused on how you accurately measure affinities of proteins. And so if you're interested, I'm happy to discuss at the end or point you to references. But what I'll tell you is that we managed to produce a panel of different peptide ligands to 2 human T cell receptors shown here. So 1G4 is a human T cell receptor that recognizes a peptide that's actually expressed in many cancers. And A6 is a T cell receptor that happens to recognize a peptide from HTLV, which is a human virus. And so the wild type peptide is shown here in red. It binds the highest affinity. And then we made point mutations to the peptide, single amino acid mutations, and then measured affinities. And we managed to produce a panel, for example, here, that varied in affinity by about 164-fold from the lowest affinity to the highest affinity. And then the plan was to simply present these to T cells and look at what kind of discrimination we could see. So to do that, we took human blood, we isolated T cells from that blood, and then we made that population of T cells, right, which all have different receptors. We basically made them all express the same T cell receptor, this 1G4 T cell receptor that we studied. And then in parallel from those same donors, we took some of their antigen-presenting cells, which we then could load with our peptides, and that's shown here by these little brown ligands. And then we could culture them together and then measure the activation of the T cell. And so here on the y-axis is a measure of T cell activation. So 100% means 100% of the T cells have become activated. 0 means none. On the x-axis is the concentration of peptide that we loaded onto our antigen-presenting cells. And then the different colors are the peptides that bind with different affinity to the receptor. And so the red one here is the highest affinity and the blue here is the lowest affinity. And what I hope you could see is that in contrast to these mouse studies, where there was a clear affinity threshold where all of a sudden there was no response, here, what we see is a more gradual loss of the T cell response. So it didn't appear as though we had this perfect or absolute discrimination that was reported in the mouse T cells. I'm going to skip this, and I'll just say that we repeated the experiments with different kinds of T cells that immunologists like to use, shown here, but the results were all the same, so I'm not going to tell you about them. But we wanted to put a number on how well these cells discriminate. And so to do that, what we did is we drew a horizontal line at 15% activation above baseline, and then simply calculated the concentration of the ligand that was required to give that response. And we chose 15% so that we could capture some of these very low affinity ligands. And so here on the y-axis, we're plotting that concentration of the peptide. And on the x-axis, we have the affinity that we've measured using a separate technique. And what we found was that if you put both axes on a log plot, you found that there was a fairly good linear correlation. So this suggested, of course, that there's a power law relationship between what we call the potency of the peptide and its affinity to the receptor. What we observed was that for the 164-fold change in affinity, we observed a change in potency that was on the order of tens of thousands fold. And so this told us that there was some kind of enhanced discrimination that the T-cell receptor was performing, because if we wrote a mass action model, the fold difference on the y-axis would also be 164-fold, similar to the KD, but it was much larger. And so we decided to simply fit a power law equation where the power here, alpha, we defined as the discriminatory power, because it's effectively the slope of this line. And if the slope is very, very steep, it means that small changes in affinity leads to very large changes in ligand concentration required to activate the T cell. Whereas a small slope, of course, means that you have less discrimination power. And that the slope we found for all of our different T cells was about 2. And you could see that this slope was significantly above 1, which is what you would predict from a simple mass action model. And so this told us that there is definitely enhanced discrimination for the T cell receptor. What was confusing, of course, was that this wasn't the case for this original mouse data that I showed you that was highly influential and, and that I spent sort of almost a decade trying to explain. And that data is shown here. And what we did is made these similar kind of potency over affinity plots. And what you could see here is that the slope in this situation is much larger. So the power here is on the order of 9 from these original studies. And and so this led to a kind of discrepancy. We were reporting a power of 2. These original studies are suggesting a power of 9. So we wanted to know who was right. And so one thing we realized is that these original studies were done in these mice that are still used by immunologists. And there's actually a lot of data that has been published since on these mice, but hasn't actually been directly used to inform on T-cell antigen discrimination. And so what we did was to simply look at those studies and try to take some of the binding data, the affinity data that was published in those studies, and combine it with some of the T-cell activation data, the potency data in these original studies. And that's what you're seeing here. We call it the revised murine data. And so when we took the new affinity measured here shown on the x-axis, using the original potency measures from these papers, we found that the discrimination power now was a lot smaller. For example, for the OT1, it was reduced to about 2, which is what we were getting. And that's shown here. There was an average of about 2.2. So it was a lot more consistent with our data. So we were quite pleased with this. And then we sort of realized that we could actually do a much larger analysis, because there's huge amount of interest in T cells, because they can be exploited and are being exploited for a variety of T cell therapies in cancers. And so people have made lots of T-cell receptors, lots of peptide MHC molecules, have measured affinities and done these potency plots. And so scanning the literature, we were able to make potency affinity plots for other mouse and other human T-cell receptors. In total, we made about 70, we made 70 potency over affinity plots from which we could calculate discrimination powers. And as you can see here, we found that the powers were a lot more similar to the values we were getting. So 3 for other mouse and about 1.6 for other human TCRs. And in fact, when we combined all of this data together, all these other receptors, we found a discrimination power that was actually, or at least the mean, was exactly the value that we got in our study with human T cells. There was obviously a large variation because these published studies weren't, their aim wasn't to calculate a discrimination power.
I had a question, Omer.
Yes, of course. Yeah.
Could you just repeat what changed in sort of this revised murine data? And in particular, do you have any hints about what might have been wrong in sort of the first analysis?
Yeah, of course. So the difference, we're pretty confident that the difference was actually in the original affinity measurements, these KDs here, which are plotted on the x-axis. And the reason for that is because these measurements were done in the 1990s using a technique called surface plasmon resonance. It's a biophysical technique that was actually just introduced in the 90s. And so I think people weren't aware of artifacts that can exist in that technique, including the protein aggregation. And so these newer measurements in these receptors were done a lot more recently. For example, for the OT1, there were some measurements done in 2014. And there were actually different peptides, not the same ones that were used in the original study. And so we actually took data from a different study here published in 2006 and actually just combined data from across two studies. And so really it's about making accurate affinity measurements on the x-axis, okay? And actually just to kind of cross the T and dot the I, in my lab, we've actually very recently decided that because the OT1 receptor is used so heavily by the community, we decided to use our new technique to accurately measure all of these affinities. And in fact, here I'm showing you the data for this OT1 receptor. There's so many different peptides because so many immunologists have used it. And so we've basically remeasured all of them. And that's what's plotted here on the x-axis. And on the y-axis are potency measurements from the published literature. And you can see here that the slope here is much lower. It's about 3. And it's the only difference is that it's a different affinity measurements on the x-axis. Okay, same T-cell activation data. Okay, so I hope that made sense. And so we really think it's just a discrepancy with the affinity. And we feel pretty strongly now that the discriminatory power of the T-cell receptor is much weaker than we initially believed it to be.
Thank you.
I'm going to skip this slide. Okay, I want to now come back to this point, which is that given that the discriminatory power of our T-cell receptor is much weaker than we thought before, it sort of made us kind of scratch our head a little bit and think, well, like, is it special? Do all receptors have a discriminatory power of 2? Because like I said before, this discrimination power of 1, which is kind of the null hypothesis, is based on a mass action model, not based on actual data for other receptors. And so other receptors like cytokine receptors, receptor tyrosine kinases, or G protein coupled receptors, these are different families of receptors. They usually don't need to bind different ligands, or certainly not discriminate between them. They generally have co-evolved with their ligands, and they bind one or very few ligands, unlike the T-cell receptor, which, like I said, needs to discriminate between a very large number of ligands that it's never encountered. But what's interesting is that for these other receptors, people have been making synthetic ligands that bind with different affinities because they're interested to exploit them for therapy. And so what we did is we found studies that did that. And then we made these potency over affinity plots and looked at their slopes or the power. And what we found was that the slope was always at or below 1 for these other receptors. And that's summarized here. So cytokine, receptor tyrosine kinases, and G protein coupled receptors all had discrimination powers that were at or below 1. And as a group, they were significantly lower than the discriminatory power of the T-cell receptor. So this kind of made us breathe a sigh of relief that the T-cell receptor is indeed very special, and it has a discriminatory power that appears to be at least 2-fold higher than other receptors. I'll skip the B-cell receptor for now, and I'll just sort of mention what this means for discriminating antigens, just going back to this problem of self-non-self discrimination. So the way that I want to talk about that is to ask what fold change in potency, or equivalently, what fold increase in ligand concentration do we need to activate a T-cell when you drop the affinity of the ligand by about 50-fold? And the reason I picked 50-fold is because that's roughly what people think might be the difference between a foreign ligand that might activate a T-cell and a self-ligand that will not activate a T-cell. And so again, if the discrimination power was 1, then all you have to do is increase the concentration of that self-ligand by about 50-fold. And then you get T-cell activation. And that would be bad because on a normal antigen-presenting cell, you could imagine that the concentration of any one peptide might fluctuate a little bit. And if the self-ligand happened to increase by 50-fold by chance, maybe because the cell was upregulating a particular pathway in its normal sort of physiological process, then that could activate a T cell and cause autoimmunity. And so this would be bad. On the other hand, T cells, on the other extreme, if the discrimination power was 9, which is what, like I said, the initial studies suggested, then you'd have perfect discrimination because you need to increase the concentration of the ligand by 10 to the 15-fold. And that's basically unattainable because the total number of MHC molecules on a cell is about 10 to the 5. So the largest fold difference you could have is about 10 to the 5. But what's interesting to us is that if the discrimination power is 2, then you need a 2,500-fold increase in the concentration of, for example, the self ligand to cause T cell activation. This is called imperfect discrimination. And so what we think might be happening is that normally the amount of the quantity of any one self ligand might be kind of low, and its expression may not increase too high under normal situations, but there might be situations where its expression might be elevated. And that to us is very interesting because the way that people usually think about T cell autoimmunity, so this is a situation where T cells kill your own tissues. This happens, for example, in type 2 diabetes, where T cells go and kill your beta cells so that you can no longer produce insulin. And normally that has been viewed as, for example, a very highly autoreactive T cell escaping from your thymus. You know, this positive and negative selection didn't go properly, and the cell escaped, and now it attacks your beta cells. What this is suggesting is that actually it could be a problem not on T cells, but actually on the beta cells, because a beta cell that might be hyper-secreting insulin, for example, it starts to, produce way too much insulin, and therefore proteins in that pathway have become upregulated, that cell might become abnormal. And as a result, T cells might be there to actually kill that cell. And so we think this is very interesting because it tells us that T cells may not only just be around to recognize foreign proteins, foreign peptides, but they might also be around to recognize abnormally expressed self proteins, that they might have that kind of discrimination as well. And there's a couple of studies that have been published to actually support that observation. Okay, so I think for the sake of time, I'm going to skip a little bit this part, and I'll just go to basically my almost last slide, where I want to talk about just mechanism. So I started off the talk by saying that kinetic proofreading was not sufficient to explain T cell antigen discrimination. I want to come back to this and ask, can kinetic proofreading explain our data? And so what we did is in the first instance, we sort of fit this model to our data, and we fit it to the potency over affinity data. And there were, I believe, 4 model parameters, and we fit every single data set that I showed you. And actually, it was slightly more data than some of the data that I skipped as well. And we did a kind of simultaneous fit of all of that data using ABC SMC. And this was done by our postdoc, Dan Wilson. And fortunately, the 2 parameters that we were most interested in, which were KP and N, were identifiable. And these parameters are effectively determine the length of this proofreading chain. So KP determines the rate at which the receptor becomes modified, and N is the number of these biochemical steps, okay? And so if you have a larger N or a smaller KP, then the time between binding and signaling becomes longer. In fact, the time between binding and signaling is simply the number of steps divided by the rate of each step. And so what we found was that the number of steps happened to be a fractional number. It was 2.67, which I have to say we weren't expecting, because these are biochemical events. So we sort of expected an integer, but we were fitting a continuous model that allowed N to be fractional. And the rate of each step was about 1 second. And therefore, if you took N and divided it by 1, you got about 2.8 seconds for the total time between binding and signaling. And so what we did is we used these parameters to basically ask, can we explain a discrimination power of 2, which of course we could, because that's the data we fit. But we also put a second constraint, which is, can we also explain the fact that T cells have high antigen sensitivity? And I haven't really talked much about this, but T cells can recognize cells that have even just one ligand. And the reason that's important is because you can imagine that if you have a very long proofreading chain, it basically reduces your ability to recognize any one ligand. And so there's this kind of trade-off between discrimination and sensitivity that has been reported for this proofreading model. And so again, the long story short is that if you're trying to get the T cell to exhibit perfect discrimination, then as you vary the proofreading steps or the proofreading or the rate of each step on the x-axis, what you find is that you can have regions where you could achieve this perfect discrimination of alpha equals to 9, but those regions of parameter space don't overlap with the high sensitivity regions. On the other hand, if the T cell has a lower discriminatory power of 2, and I should say that this is the kind of thinking that went into excluding kinetic proofreading as being sufficient to explain this high discrimination power that I showed at the beginning. And this kind of motivates adding this mechanism X. On the other hand, if you're asking, can kinetic proofreading explain a discrimination power of 2 and achieve that level of discrimination, but also high sensitivity, what we find is that indeed, there is a region of parameter space where both can be satisfied simultaneously. And in fact, the numbers that we get from our data for the number of steps and the rate of each steps actually fall really here at the bottom. It seems like the T cell is kind of is tuned to have the smallest number of biochemical steps possible to achieve both high sensitivity and this level of discrimination. And so the conclusion of this slide is simply that we believe that this kinetic proofreading model is sufficient to explain how T cells discriminate antigens. And what my lab is doing now is trying to identify the molecular steps that, the actual molecules in T cells that mediate these biochemical steps, and then trying to manipulate them to kind of see if we could increase or decrease the discrimination power of the receptor. Okay, and so I'll just end by concluding and saying that the T cell receptor exhibit enhanced but imperfect antigen discrimination with a discriminatory power of about 2. This discrimination is higher compared to other surface receptors, which appear to have a discrimination power of at or below 1. The T cell receptor, we believe, exhibits the operational model of kinetic proofreading. Okay, so somehow superimposed on its complicated signaling cascade, there is kinetic proofreading operating there. And it has a time delay of about 2.8 seconds between the ligand binding and the receptor signaling. And it induces this delay by having this fractional number of steps, about 2.6 seconds. I haven't mentioned the fact that there's other receptors on T cells that also can impact the discrimination power, but they're there. And that T cells can actually become activated to very low affinity ligands, for example, self-ligands, provided they're expressed at ultra high concentrations. And so there's some implications for this, including the possibility that we might be able to improve the discrimination power of T cells. And we're doing that, again, because we studied all these mechanism X that can improve discrimination beyond kinetic proofreading. We're trying to see whether we can engineer T cells to have some of these mechanisms in order to improve their discrimination power. So I've already thanked the people that did the work, and I just want to thank all my lab for lots of discussions on this project. And my collaborators locally are Anton van der Merwe, Simon Davis, Michael Dustin, Marion Brown, and Oreste Acuto. Sam Isaacson was involved in some of the data fitting that I shared with you today, and we're funded by mostly by the Wellcome Trust, but also from CRUK and from another local trust here in Oxford called the CIU. Okay, so thank you.
Well, thank you very much for such an interesting talk. So please feel free to ask Omer questions if you have any.
I had one question yeah. So one thing that I'm quite curious about, I mean, it seems like you're kind of taking into account this extreme sort of sensitivity of the T cell and the ability to detect even a single one of these APCs. And so I'm just curious, I mean, on the one hand, it seems like one way of kind of increasing this kind of discriminating power, maybe either in addition to or instead of kinetic proofreading, would be if it could sort of detect multiple ones at the same time.
Yep.
And I'm probably getting the terminology all wrong, but that was kind of one idea. And then the other question is there any chance that actually the opposite is happening, where the T cell is more sensitive to those cells that have fewer of a specific kind of antigen on them?
Okay, so to answer your first question, I think so I think having more ligands or even sampling the same ligand multiple times, I think can increase the confidence that the T cell has in that it's recognizing a higher affinity interaction, compared to these self ligands that might just by chance have bound for a long time, right? I think the T cell is probably doing that because these interactions, the binding kinetics are very, very fast. So, the bond only lasts for about a second, but actually you need to wait like minutes to hours for the T cell to become activated. And so we do think that there is this kind of sampling that happens in this reinforcement. I'm not sure if you could write down a model that can do that, that's not based on kinetic proofreading. I mean, we've tried, but I think it always comes back. You can always map it to a proofreading model at some sense. Yeah. And then the second question was, is it possible that T cell is more sensitive to cells that only have the self-peptide MHCs? Is that right?
So I guess because you mentioned that, for example, in this case of HIV, it's actually quite few ligands that appear. And so then what I'm imagining is some infected cell that is mainly composed, all the ligands are kind of self ligands, and then you have very few of these sort of unusual HIV markers. So I'm just curious if that sort of sparsity could be something that actually the T cells are selecting for in a way.
I'm trying to think. I mean, so if you were the T cell and you're using your receptors to kind of touch that cell, I think you wouldn't know that there's few foreign antigens in advance. I mean, the only way you'd know that is by whether or not they bind to your receptor, I suppose. And so there's no kind of, the APC here, I guess, if there was a way it could sort of help the T cell and say, hey, look, I have few of these foreign antigens, check me out. But the APC is often quite agnostic, and viruses, they kind of prevent the APC from raising flags to T cells in many ways. Yeah. So the T cell kind of doesn't have an option. It has to kind of, it has to tune its sensitivity to be very, very high. Yeah. Yeah, great.
Do you have any questions from the audience? If not, I wanted to ask Omer a question. So in all those models, are you sort of incorporating the spatial movement of those receptors? Could those be some sort of another factor that leads to like this enhancement that you observe?
Yeah, so it's a good question. So, I mean, all the data that we get is input-output data. We sort of measure the activity of the T cell post-transcription, and we and the input is we vary the ligand dose and affinity. So we treat the T cell as a kind of black box. Mm-hmm. So we don't have any spatial information. And so what we do is we just fit the simplest ODE model, which is basically kinetic proofreading and nothing else. And we kind of ignore all of the complicated signaling in the cell because again, we don't measure that. So one can ask, how is kinetic proofreading, for example, implemented at a molecular level, and does the spatial distribution of receptors, is that important? And the answer is probably it is. And people are measuring sort of what happens using single molecule microscopy. And they're looking at the spatial distribution of the receptor and its movement. And in fact, this is something Sam is actually like interested in, of course. And he's getting actually, he's interested in, and people generally are doing this. They're trying to get very accurate topologies to simulate diffusion on and seeing how that might impact some of these biochemical processes. Yeah.
Thank you. Any other questions for Omer? Alrighty, I think we can wrap it up here for today. I would like to thank our speaker Omer again and everyone for your support and participation. So we will take a break for the Thanksgiving and resume one last time after the Thanksgiving. So hope I will see you all back then. See you guys.
Thank you.
Okay, bye.